My name is Eric Miller. I'm a graduate student in the lab of Dr. Hadley Sykes, and together with my fellow graduate student, Kiju Sung, we're working to kind of reinvent diagnostic tests, and specifically by developing new minimalist binding proteins that we can use to replace the antibodies that are commonly used in these diagnostic tests. So when we think about kind of the classic immunoassay, the classic diagnostic test, the thing that might come to mind as the thing we're most familiar with as consumers is a pregnancy test. And this is kind of the archetypal format where it has a support, a nitrocellulose membrane, and on that support we have an immobilized antibody. This is a capture protein, a binding protein that can grab onto another protein that is a diagnostic biomarker. So this is a protein that's associated with some sort of condition, in the case of a pregnancy test, pregnancy. And so now that we've captured this protein out of a patient sample, we need to go ahead and associate some sort of detectable signal with that capture event. And so we have a second antibody, a detection antibody, and we have a label that's associated with that. So in the case of a pregnancy test, we literally have the concentration of millions of gold nanoparticles that are in this thin line, and that's what allows us to see it with the naked eye. So we know that this test format works. And in fact, this sort of format can be applied in any sort of diagnostic test where you have an identified biomarker that you can detect in a patient sample. But I'm here today to propose to you that the antibodies that are used in these tests are not ideal for this sort of application outside of the body. So these proteins are large, they're complex, and as a result, we can't produce them in bacteria. Oftentimes, we have to produce them in animals, so we'll inject our biomarker into an animal and then get it to raise an immune response and harvest the antibodies from its serum. That's a long process, and it's also expensive. Additionally, these antibodies are thermally unstable. So studies have conducted accelerated degradation tests where you basically expose these antibodies to elevated temperatures for extended periods of time, and you look at if they retain their activity. And at 60 degrees Celsius for only 5 days, we see that these actual antibodies used in malaria diagnostics break down altogether. So if you're looking to make diagnostics that are applicable at the point of care, you need for these to be thermally stable. Additionally, these antibodies are difficult to engineer for new properties. Because they're so large, we can't really manipulate them on the genetic level, on the DNA level. And so instead, we can only modify them chemically, which limits us to reagents that are available commercially. And then additionally, in the process of modifying them chemically, we have losses to process inefficiencies. There are a couple of other things. So they are prone to off-target binding. The actual functional part of the protein is relatively small, and so there's a lot of footprint where you can have non-specific binding of other proteins that can lead to false positives or false negatives in your test. There's lot-to-lot variability because you're relying on animals and their immune response to develop these reagents, and there's a restrictive IP landscape. So we think these are all critical problems that are facing antibodies, and we want to address all of them. For this talk, I want to focus specifically on these top three. So we're looking for a new sort of protein we can use in the place of antibodies that is both cheap, that is stable, and that we can manipulate and engineer for added functionality. And so we thought that we might kind of take our cues from nature, and so we look for proteins here in hot springs. And specifically in this bacteria, which is a thermophilic bacteria that lives in these hot springs, and specifically at this protein, SSO7D. This is a DNA binding protein natively, so it has this function. But we know that because there's a defined binding face that's involved in that interaction, we can tailor that. We can basically swap that out in a modular fashion to repurpose this protein for our targets of interest. Additionally, because it exists in bacteria natively, we can produce it cheaply in bacteria. Because it exists in hot springs, it's intrinsically stable. And then because it is so small, it's about 1/20th the size of antibodies, we can manipulate it really readily. And so we wanted to validate that this was a good candidate for these applications. And so we found that, in fact, SSO7D is cheaper and faster to produce than antibodies. So if we pit these in a race against each other, SSO7D, because we can produce it in bacteria and because it's very small, we can make a lot of it very quickly. So in the time that it takes to make 1 test's worth of polyclonal antibody material, we can make 100 tests worth of SSO7D. And this is at a cost of 2 cents, whether it's 100 tests or 1. So if it's me, I'd rather have 100 tests. In fact, for SSO7D, in 24 hours, we can produce enough material for 170,000 diagnostic tests at a total cost of about $40. And that's without pushing the process at all, in a flask about this big. So it's cheaper, faster to produce. Additionally, it's more stable. So if we run our own accelerated degradation study at 95 degrees Celsius, practically boiling the protein, we see that antibodies lose their activity very quickly, so within about a minute. In contrast, SSO7Ds, again, practically boiling these proteins, retain their activity all the way out to an hour and a half, 2 hours, and it's about a 275-fold improvement. So obviously, in the field, we won't be boiling these proteins, but this difference that you see in the stability is a huge improvement in terms of the diagnostic stability and the shelf stability of these tests. Additionally, SSO70 is more versatile than antibodies. So because this is very small, we can actually manipulate it and tailor it on the genetic level. And so if we wanted to add functionalities to it, fuse it to other functional proteins, we can do so really easily. So if we wanted to, say, attach it to a color-producing enzyme, or a fluorescent protein that we can see with a UV reader, or say an anchoring functionality so that we can put a lot of this protein onto a surface, we can do that very readily. So our first applications that we're looking at, two kind of disparate fields. We're looking at traumatic brain injury and aquaculture, specifically infections in fish stocks. The reasons for these, they have similar dire consequences and a huge scale, but the huge thing is that the diagnostic methods that are available for these applications are both time and labor intensive. And so we think that our application can be used in the place of standard antibodies. So our new and improved immunoassay would use these SSO70 molecules in the place of those antibodies, specifically because they are cheaper, quicker to produce, they're more suitable for use in harsh environments, and because we can engineer them readily for different applications. Thank you guys for your attention. If you have any questions, I'd love to talk to you at poster 13.